We present a protocol to prepare free-floating slice cultures from brain tissue collected from living human donors submitted to resective brain surgery. These cultures are amenable to perform biochemical and cell biology assays or immunohistochemistry. It is expected to contribute to the elucidation of the mechanism underlying neurodegeneration in age-associated brain diseases. The main advantage of this technique is that it is a simpler and cost-effective alternative to the classic slice culture protocol using membrane inserts. Although the overall protocol is not complex, some steps, such as removal of meninges, gluing the tissue to the vibratome specimen disc, and resection in slices for immunohistochemistry, are best understood when demonstrated visually. Helping to demonstrate the procedure will be Niele Mendes and Glaucia Almeida, who are grad students, and Giovanna Nogueira, an undergrad student.
To begin this procedure, add salt to a bucket of ice. Transfer the slicing solution to this bucket and let it rest under carbogen mixture, bubbling for at least 20 minutes prior to use. Next, prepare a block of 3% agarose that is about 2 centimeters by 2 centimeters by 2 centimeters. Superglue the agarose block to the vibratome specimen disc in order to create additional mechanical support to the tissue sample during slicing. On the vibratome, set the section thickness to 200 micrometers, the vibration frequency to 100 Hz, and choose a slicing speed between 0.5 and 1.0 millimeters per second.